There we are.
Great. Thank you, Robert. Yep. So again, welcome. We're here for the QAAFI Science Seminar. Today, Robert Henry is going to give us a talk about recent advances in technologies and applications of genomics in plants. Next slide, please. And so my name is Pauline Okemo. I'm a postdoc research fellow in the Center of Crop Sciences. I work with Robert Henry, and I work in the genomics and gene editing platform within his group. Next slide, please. So we'll begin by acknowledgement of country. UQ acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. Next slide, please. So a little bit of housekeeping. The seminar is going to run for 1 hour between 12 to 1. And so we'll give Robert time to give his talk. And at the end, we'll have a Q&A session. So there's a Q&A tab. So remember to type in the questions that you have, the Q&A tab rather than the chat function. And so we'll have a bit of discussion at the end of Robert's talk. Next slide. So without further ado, I'll introduce today's speaker. Today's speaker is Professor Robert Henry. Robert conducts research on the applications of genomics in the development of new products from plants. He's a professor of innovation in agriculture and was a foundation director of QAAFI, which is Queensland Alliance for Agriculture and Food Innovation, for those that are new here. He works in the ARC Center of Excellence for Plants, Success in Nature and Agriculture, and is the director of the ARC Research Hub for Engineering Plants to Replace Fossil Carbon. So Robert is going to tell us about how plants can make us millions and millions of dollars. So take it away, Robert.
Thanks, Pauline. And I too would like to acknowledge the traditional owners of the land on which we meet. Today, I'm going to take the opportunity to tell you about recent advances focusing on particularly the things we've done in the last year in terms of the technologies we're applying to genomics and some of those applications and some of the important outcomes or discoveries that have resulted. So in the last year, the technical advances have really allowed us to assemble and annotate genomes on a scale that really we haven't been able to do in the past. We've been able to tackle successfully polyploid genomes, and that's something that wasn't possible not too long ago. We've made some real progress and gained insights by looking at long-read transcriptomes, and we're heading towards the exciting new area of spatial transcriptomics. So I'll give you an introduction to those and some of the applications. We've applied our technologies to many crops listed here, and many of the crop wild relatives that we're looking to use as ways to diversify or expand the genetic base of these crops. And we've also applied genomic technology to conservation of natural environment and biodiversity, both in natural environment conservation, but also in understanding how to manage rare and endangered species. The crops that I talk about, mango, citrus, macadamia, rice, banana, pineapple, custard apple, papaya, passion fruit. We've sequenced all of these to produce high-quality reference genomes, and we've also tackled some of the polyploids, coffee, sugarcane, channel millet, and triploid banana. As a bit of introduction, some of the horticultural genomics we're doing has been funded by Hort Innovation, and I acknowledge the two major projects at the start here. Our horticultural tree genomics program that looked at the 5 groups at the top, and the more recent 5 at the bottom that we're now moving on to, funded in the program to develop a platform, a genomics platform for horticultural industries in Australia. The first example of I think, what is a real milestone in plant genomics is our chromosome level sequencing and assembly of the mango genome. And the reason that this was remarkable is that we're able to do it from the sequence data alone. In most cases, or in all cases that I'm aware of, people have relied on some knowledge of the genome, a genetic map, some other tool to assist with the assembly. Here, just taking the reads from the sequencer, we were able to assemble the entire genome telomere to telomere. And so that really is a milestone in that the technology's got to the point where we can do that. We're not always going to be able to do that, but it just shows how much easier producing the sequence of a plant is. And there's been many applications of this. We've applied this working with people like Heather Smythe to understanding some of the genetic basis of differences in mango flavour and quality. And Melanie Wilkinson has really used this to understand something of the genetics of this organism and to explore things like some of the chromosomal inversions that are really important to understand in plant breeding. We've applied this technology very extensively to the citrus species and all of the Australian wild species now, which represent around a third of the genus. We have high quality reference genomes for, but we've also applied it to understanding and gaining sequences of domesticated citrus. And for the first time, we've produced a de novo genome sequence of a variety produced by mutagenesis. So traditionally, we produce seedless fruit by mutating an individual. And here, for the first time, we can see what that impact has been at the genome level. And we may have altered lots of individual genes, but what we've seen is substantial changes in the structure of the genome. What the mutagenesis is doing is rearranging the chromosomes. And I think that the seedlessness in these fruit can be explained by the inability of these lines to actually form viable pollen because they can't, chromosomes can't pair because of structural rearrangements. And this may be a common mechanism by which we routinely achieve seedlessness and that it can be really seen at the genome structural level rather than at just the individual gene level. The Australian citrus have become very important in the world because of the citrus greening disease that is wiping out citrus in the Northern Hemisphere. The price of orange juice has increased because of the shortage of citrus in all the traditional production areas. These Australian wild species have resistance to this disease, and that's being used very actively by citrus breeders worldwide. And now, of course, our genomes reveal the location of the defense genes throughout the genomes of these disease-resistant wild relatives. There's a great example about how genomics is facilitating the use of crop wild relatives to rapidly breed to cope with changes in the environment, in this case, the emergence of a major new citrus disease. The macadamias are very important to us in this part of the world. These are the 4 species, and we've got now really high quality reference genomes for these, and we've sequenced the germplasm that's used extensively in the macadamia breeding programs. What we've identified is that these genomes are a result of relatively recent whole genome duplication, and that a significant proportion of the genes are duplicated on another chromosome. And this is very important to know if you're going to use molecular techniques in breeding a species. But probably the most striking outcome of the macadamia genomics program has been that we've identified extensive reticulate evolution. These 4 species are very good species in a cladistic sense at the nuclear genome level, but there's been extensive exchange of chloroplasts. And this is a phenomenon we see in almost every plant group that we look at, that reticulate evolution is really important in understanding the origins of many of the plants we work with, that we don't just get divergence in a traditional evolutionary tree, but there is gene flow between the branches due to rare hybridization events. And here in macadamia, we can see a long list of history of of evidence of this reticulate evolution here in southeast Queensland and northern New South Wales that hasn't perturbed the definition of the 4 species, but it's allowed chloroplasts to be shared locally between the species, something that challenges traditional species concepts for some people, but really is important to understand if you're working with this material and breeding from it. One of the more recent genomes we've sequenced, not yet published, is the pineapple genome. And here we have a haplotype resolved genome. And this is the next important point I wanted to make. It's really critical that we have haplotype resolved genomes. Even for humans, only a few years ago, we talked about 1 sequence, when in fact we know there are 2 copies in a diploid organism. And those 2 copies, maybe in humans, are very similar. So the single sequence is reasonable. But in many plants, it's not reasonable to work with a single sequence because the 2 haplotypes are incredibly different. And that's very much the case here in this pineapple example. And here we're comparing the 2 haplotypes at the chromosome level. And you can see in the orange, those areas are places where there's been inversion of the sequence in one haplotype relative to the other. And it's most evident in some of these other chromosomes. You'll note on chromosome 24 that there's a huge part of the chromosome that's inverted. Now, we've checked all this very, very carefully, and these are real inversions between the haplotypes. And you'll notice on chromosome 20, that region that's been inverted has quite a large number of genes. What this tells us is that these areas are areas where there can't be recombination in meiosis because the chromosomes can't pair. So these genes in these regions are going to be inherited as blocks. And so we're working with Garsinuski, the breeder, to understand which of the traits in pineapple are likely to be in these blocks that really can't be broken up, certainly by working with this frontal line. Now, this is the smooth cane variety that's been favoured in Queensland for canning, and this is a pre-Columbian variety. So this variety arose probably thousands of years ago and has been cultivated ever since, preserving a remarkable degree of heterozygosity. And you can see structural differences that are very extensive between the haplotypes. And this heterozygosity may be part of the vigor of this plant and may be critical to its performance, but it has great implications for any breeding. And so we see similar results to this in many of the species we're working with, and these insights are really critical to support ongoing genetic improvement. But we've also looked at species that have been sequenced before. Rice was only the 2nd plant genome to be sequenced, but we resequenced it again using the modern hi-fi sequencing technology, much more accurate than the sequences that are available online, the references that people have been using up until this year. And what we found is that we can identify about 10% more genes in the more accurately sequenced rice genome. Now, this is quite remarkable for a species like this that has been such a central species in plant genomics that really the annotation of the genome requires an almost perfect sequence because the slightest variations prevent reliable annotation. So what we recommend from this is that if we're working with species that have been studied for a long time, we really do need to revisit the genomes with modern technology and upgrade the quality of the sequence. And of course, it's very easy to do that with current technology, and it's really worth the effort rather than just using a reference genome that you might find online. We work very much with wild rices, and Australia is a very important part of global rice biodiversity, and we've formed this year major international collaborations that are involving representatives now of very large numbers of countries in which wild rice is found, trying to coordinate the efforts to conserve the genetic base of this most fundamental crop. Some of the Australian species are really important, and we've sequenced those to produce high quality reference genomes in the last year. If we look at Oryza meridionalis, this is the Australian endemic species that really is basal to the clade that was domesticated in Asia. And we see very significant chromosomal differences here that are probably quite important in understanding the last steps towards domestication of rice, and many really important adaptive traits that we can see in the wild species that may have value if introgressed into the domesticated gene pool. So rice heredity analysis has been the subject of quite intense study, and we're embarking on a de novo domestication of this species by editing domestication loci. Rhiza australiensis also has very novel characteristics, a larger genome, 909 megabases, fairly high repeat content, and what's remarkable here, I suppose, is that we're able to get such an accurate genome now for such a genome with 75% repeat content. This is the largest of the rice wild relatives. So working with crop wild relatives more widely, many things have arisen. One, what I'd like to note is that this grape here is a wild grape, is a local species from the area around Brisbane. Many years ago, DNA analysis indicated to us that this was much closer to domesticated grape than the Cissus genus in which it had been placed. Very pleasingly this year, it's been reclassified into a new genus, Apocissus. So this is Apocissus antarctica, a result of genome analysis. And so we are driving the taxonomy to reconsider what's out there and really characterizing a very important local wild relative of grape. So our genomic evaluations looked at the wild millets, the macadamias, the citrus, the rices, and is going on. In sorghum, we have a major program now in collaboration with JGI and Monash University. And here we're sequencing effectively the wild species. Sorghum is essentially an Australian genus with a few species that extend outside, was domesticated in Africa, but really all of the diversity is here in Australia to be characterised. Many of the species are not well known, occurring in Queensland and the Northern Territory, but we now have high quality reference genomes for these. And these species have some amazing characteristics. There's groups with 5 chromosomes and others with 10. And the latest results are showing that the smaller number of chromosomes have arisen by chromosomal combinations. And so we've got a very unique mechanism here of evolution of the species within the genus. And many of these species are now going to be useful sources of genetics. One of the big developments has been the production of a mutant population. This has been funded by the Carlsberg Research Laboratories, where we've produced 155,000 mutant lines of sorghum. And these lines exist in the cold room and as DNA. So if we choose to find a mutation in a particular gene, we can screen that using the Carlsberg method of digital drop PCR that allows us to find a mutation in every gene that we've looked for so far, and within a matter of days be growing the plant to see what the phenotype is. A great tool for gene discovery in sorghum, and it's something that we believe will be an important tool for plant genetics generally. We've worked quite a lot with the polyploid species, and these have been really hard in the past. A great example of that is that back in 2014, we were part of the consortium that sequenced the Robusta coffee genome when we published that in Science. But it was 10 years later, this year, before we were able to publish in Nature Genetics the sequence of Arabica coffee. Arabica is a tetraploid, and it is much more difficult to sequence polyploids, or it has been. But really, this is a sign that the technology has caught up with the challenge of working with complex plant genomes. And the ultimate example of that is sugarcane. We've published books and wrote reviews on the challenges of sequencing the sugarcane genome. It really has been the ultimate challenge for plant genomics. Having a very large number of chromosomes, being a dodecaploid with 2 ancestral subgenomes and recombinations in the chloroplasts. We have different chromosome numbers in different genotypes. But finally now we have published in Nature this year, finally a high quality reference genome for this, the last of the 20 major crops in the world to have a reference genome, and certainly the most complex, with 150 or so chromosomes in a typical variety. Adam Healy, the senior author, did his PhD with us here in QAAFI a few years ago, working on eucalypts. The CIs in this project were Karen Aitken from CSIRO and Angelique DeHont from CIRAD, to acknowledge them, and myself. We were very fortunate to be in the last phase of putting this genome together. But there are other examples. Rahul, a PhD student, now supported by Indian Council of Agriculture Research, working with us in QAAFI, has produced a high-quality reference genome of channel millet, Echinoclea turneriana. This is one of the species we're working with the Mythica people from the Channel Country on. We believe is one of the traditional foods, and a remarkable species with great agronomic characteristics. It's a dodecaploid, and it is quite remarkable to find such a high level of ploidy in a naturally occurring plant. Sugarcane's a human creation. It wasn't naturally a dodecaploid, but this is, and we've got a remarkably good genome, haplotype resolve finally of 106 chromosomes. Something that really is probably in some ways an even greater triumph than being able to do this with sugarcane. We hope to learn a lot more about the biology of this species and its potential to be a millet that would be adapted to a wider range of environments. I'd just like to also mention the work we've done with conservation support. We work very closely with the Sydney Botanic Gardens. I'm involved in their advisory board, and what we've done this year is sequence with the support of Bioplatforms Australia, Threatened Species Initiative, the Idathea hardeniana, the nightcap oak. I remember when Robert Koyman discovered this species in 2002, just over the border in New South Wales. A species that's very rare, less than 100 individuals, and many of those destroyed by recent fires. This genome is a good example of one that now probably the best in this plant family that we've been able to put together, that allows us to sequence all the individuals of a species to completely define its genetic variation. This species is a good example of where we've been able to use to complement our hi-fi PacBio sequencing, which normally produces very complete genomes, to close the remaining gaps by using Oxford nanopore reads above 50 kb. So using the very long, only the very long ones. The short ones don't help. It's got to be just the long ones. So we've got some interesting interactions of technology in putting together some of these complex genomes. I'd mention that Patrick Fay, who's been a postdoc in QAAFI based at the Sydney Gardens, has now moved to the Herbarium in Brisbane to lead genomics there, and will continue working with us probably on projects on the genomics of the wild soybeans, Queensland being again the centre of diversity for this genus. Richard is a PhD student with us, and he's working on using genomics to guide restoration of rainforests. We now screen the planting material and source diverse material to ensure that 95% of the allelic diversity in a species is included in the plantings of any restorations of rainforests. This is going a long way to ensuring that we maintain biodiversity in any conservation work in planting of natural areas. I just want to turn to transcriptomics because we do a lot at the genome level, but at the transcriptome level, there are really new insights that we can get because the technology is driving that. And long-read transcriptomics we've done in rice and in many species. The one that I'll talk about today is sugarcane because it's so complex. And I've talked much about our short-read sequencing there that's allowed us to look at the biased expression or failure of bias in expression of different alleles within the genome. But we've been able to look at the contributions of the different subgenomes in sugarcane. Because we can, with a long read sequence, we can distinguish the transcripts that are coming from each of the subgenomes. We can look at the relative contributions of the different chromosomes and subgenomes to traits that are important, or the expression of genes that are important in traits like the sugar content and other things that we're trying to work with in sugarcane. This complex genome here, we can look, this is depicting the subgenomes and their comparison. And what we were able to do by working with a number of international colleagues in India is to find an example where we had a hybrid, modern hybrid sugarcane, where we knew the identity of the varieties, the varieties of that contributed their subgenomes. So we're able to sequence the transcriptome of the progenitors and the hybrid, and then attribute the expression. And it did confirm that the genes controlling sugar content being expressed in modern varieties are coming from the Officinarum subgenome, and many of the stress and disease resistance type genes are coming from the Spontaneum subgenome. What this does is give us the potential to redesign sugarcane with different chromosome composition that might have a better combination of the traits that we're looking for in an improved sugarcane variety. The other thing we've discovered is that the long transcripts are not well represented in the literature. Most published transcriptomes are very poor in the long read transcripts, and what we found we need to do is specifically target sequencing long transcripts if we want to see them. And this has effectively doubled the number of transcripts we can find by better representing those really long transcripts due to long genes or long versions that were missing in all of the analysis to date. Quite an important improvement in the technology. What this allows us to do is look at the multiple transcripts, the 4 or so different transcripts we might find from any one gene, and look at the frequency of things like intron retention, which we find is remarkably widespread in plant transcriptomes, and differential splicing of all types. Now, one of the really exciting areas of technology today is the fact that we can do spatial transcriptomics, and we've started to dabble in that in wheat and dragon fruit, and spatial transcriptomics allows us to look at which cells are expressing where in the plant genes are being expressed, and that's quite important in many of our breeding efforts where we want to change genes in a particular part of the plant to modify a specific trait. We've used a range of technologies in the work that we've done, but the one that we're focusing on particularly at the moment is the stomics method, where we use an array of 4 million oligos per square centimetre. We section the tissue, and by touching that on the array, we can link the RNA in the tissue to oligos that are sequenced to have effectively a code that when we sequence the RNA tells us where they were in 2 dimensions on the surface of the tissue, and we can place all of the transcripts back into the individual cells in which they were found. So we're doing this in the developing wheat seed, trying to understand the impact of stress on developing wheat and the implications for yield. So this is a microscopy of the developing seed, and here we have total gene expression. This is work by Tori Milstein. Tori's a PhD student that's working on this, and Tori's now working to categorize the different classes of genes expressed in the different tissues, and on looking at the individual genes in terms of the cells in which they're expressed. A key application for that for us is to understand in the pericarp the roles of various metabolic processes in the endosperm and pericarp in determining grain quality and grain yield that influence the rate of grain filling, and we hopefully will be able to soon describe gene expression at the individual cell level across the developing seed. One area where we did successfully apply it is with Jin Lee, a visiting Chinese scientist working with us in QAAFI, working on dragon fruit spatial transcriptomics. We chose the dragon fruit because it has very distinct tissue types within the developing fruit, and we can look at, in this case, trying to understand what determines shelf life of a fruit, what genes are expressed that control the senescence of the fruit. And this has been very successful in that we can compare the exocarp, mesocarp, and endocarp parts of the in terms of cell-specific expression, and we can look at single cell and spatial transcriptomics together. In this analysis, we looked at using some great metabolomics technology to look at 3,000 metabolites, and what we've discovered is regulation of the early stages of senescence. We've known for a long time in fruit about the roles of ethylene and various other things in senescence of the fruit. But what we haven't really understood is the master switches or key technologies that initiate that process of senescence. And now we've got evidence for new candidate genes that might be really valuable in extending fruit shelf life. And this is something that can only come from that spatial analysis that we can now do about knowing where in the fruit and when genes are expressed. One of the interesting applications of our current technology is we're trying to understand, coming back to answer some of the big questions in genomics. We're looking, and this is some of Abby's work, where we're looking at the evolution of genome size. In the Proteaceae family, an Australian family, we've got around 100-fold difference in genome size between one species and the next. We've got species in the northern rainforests that have chromosomes that are bigger than the human genome. And we have others that are quite small genomes, all in the same plant family. What's driving that? What we've done is sequence across the diversity of this family and are looking to try and understand how that's evolving and whether it relates to the environments in which the species are growing. It still remains a great mystery why we have such a huge range of genome sizes in otherwise closely related plant species, and we're trying to understand if it has any adaptive significance. What I'm aiming to support in this work is the ability to capture more of the biodiversity in the wild by introgressing novel genes, and that's much easier when you can see everything. But also in thinking about de novo domestication. Here we've got from the Channel Country a cereal and a pulse that seem to be really great candidates as new crops, opportunities to diversify the genetic basis of food production and adapt to the inevitable impact of climate change on the need for more resilient crop species. So I'll end there by just acknowledging that there's many people been involved in this, and one of the achievements I think that's really interesting to me is that the new AI generated metric of Scholar GPS has ranked Agnello and myself as #1 and 2 in the world in terms of research on the transcriptome. That's across all organisms in the last 5 years. And I think that's because we've worked with so many systems and really made such significant advances that indicates, I think, that working at the transcriptome level is something that we really have to focus on and can continue to focus on into the future. It's important to acknowledge the research funders, and there's going to be many more organisations joining us in the new research hub over the next few years, but these are all the major contributors to the work that I've been talking about today. But I'd be very keen to entertain any questions and discussion about this work. Thank you.
Thank you so much, Robert. It was a great talk. We do have some questions in the Q&A tab, and one of them is from Andres. Considering the reducing costs of quality producing genome assemblies, Will it be feasible soon to produce individual assemblies in genotyping, population genetics, GWAS, would be replacing SNP databases?
Yeah, so one of the areas I'm enthusiastic about is using genome sequencing as a core genotyping tool. I believe that genome sequencing, with the convergence of marker technology and sequencing technology, should be the preferred tool for all analyses, that we probably should be thinking about capturing the whole genome for every sample. The cost now is becoming very competitive. The major cost of doing genomics is going to be collecting the sample and extracting the DNA. Once you've got that, I think the opportunity is wasted if you don't sequence the genome. What we're seeing is, and we've got quite a few efforts going on to look at how we can do that at minimum cost. For many of the species we're working with now, it costs less than $50 to do that. We're looking at targeting getting down to around the $10 to get a really high quality genome sequence. But one of the really exciting areas that we're working on is long read sequencing for genotyping. It might seem counterintuitive, but what we're finding is that the long read sequencing is proving to be a great genotyping tool. Because each of the reads can only map to one place in the genome, we don't have the loss of data that we have with short read sequences because of the potential to map at more than one location. And we've got some great examples now in complex genomes of using long read sequencing as a genotyping tool. Again, the effort has to be on getting down DNA isolation costs and library preparation costs. The sequencing cost is not the main cost here. And so I'm looking forward to some exciting developments there. And in the next few months, we've got a number of workshops on how to get long read sequencing to be a cost-effective tool in this area.
So, another question from Michael Woodvardy says, great talk, Robert. The question is, to what extent, if any, does intron retention in transcripts simply reflect intermediates in mRNA processing?
It's a good question. I think, are all those transcripts that we find being translated into protein? I guess we don't know, but the abundance of them suggests they probably are. What makes intron retention real for me is that when we find introns that are retained, they're essentially always have a multiple of 3 nucleotides. So what that suggests is that they can be translated to produce a protein, and that they don't disrupt the function of the protein other than inserting a whole lot more amino acids. So that makes me think that most of these are real, and we have to think about what those different functions might be of those different versions. What we're, of course, very interested in is whether there are any differences in the transcripts we find in different cell types or in different tissues, and whether this is part of the regulation, targeted regulation of gene expression, and what mechanisms there would be that control that.
Thanks, Robert. A question from Peter Waterhouse. Are there any patterns in the types of genes that are found by long read sequences that were overlooked in pre-long read sequencing?
Well, I think it's clearly genes that are longer. So it's if bigger genes encoding bigger proteins are underrepresented in most transcriptomes, because even though you're using long read sequencing, the libraries are being biased towards the shorter transcripts that are there. So if you've got a gene that encodes a large protein, it's much less likely to have been represented. And also, if you've got an alternative splicing of that leads to a larger transcript, it's less likely to have been picked up in the past without producing libraries just of the very long transcripts, which is what we do to correct for that inadequacy.
Right. A follow-up question from Peter again. How much of the differential splicing is creating different functional proteins versus mistakes in splicing versus expression regulation?
I don't know, Peter. It's a good question. It'd be great if you can help us answer that. Um, uh, all we can see is that they're there, and it may well be that some of those are mistakes, and so they're wasted effort. They don't actually impact on anything. But I think the intron retentions are quite often real.
There's an interesting question here, Robert. Thanks, Robert. How can one access the EMS sorghum lines for research?
Contact us. We do have to negotiate with Carlsberg about these. Carlsberg has some patents in this space, but we have the populations here in the cold room. The populations are duplicated in Copenhagen and Brisbane, and the DNA is mainly in Copenhagen. But talk to us. We can work out how to access that.
All right. A question from, we still have some time. We have more than 10 minutes. So there's a question from San who says, hi, Robert, great work. I'm interested in genomics in passion fruit. Could you talk more about current work or any future plans you have in passion fruit?
Yeah, so we have, this is Mo's breeding program. QAAFI's just taken on the passion fruit breeding program, and we now have people like Patrick Mason joining that program. I can see that it's going to, it's very early days. We have our first genomes, but there's a very active program now because of the resources here in Queensland and at Murdoch being directed towards passion fruit genomics, that we're going to see a whole lot more resources available to, one, understand, of course, the biology of passion fruit, but two, of course, support the breeding of what's been a relatively minor crop, but might have the potential to grow with greater science input.
Great. So just a reminder, we still have around 5-7 more minutes. So if anyone has a question that they would like to ask, please put it down in the Q&A section and we'll forward it to Robert. So one more question, any workshops planned related to transcriptomic data analysis?
We aren't planning any at the moment, but I have to say there are a lot of them about. So if you look online, a lot of people are running transcriptome analysis courses and workshops, and the techniques that are being used are evolving quickly. Conventional RNA-seq type analysis, that's advanced quite a lot in the last few years, but where we're seeing the great explosion, of course, is in spatial transcriptomic analysis, where we'd expect the tools to keep evolving very quickly, given this is such a new and emerging field with so many exciting results coming out. If we look at spatial transcriptomic, it's been used mainly in animal systems, and it's only in the last year or two that we started to see any reports in plants. And so we will be thinking about, particularly, I guess, if there are any unique aspects of analysis of spatial transcriptomes in plants. Getting sections of plants is more challenging, but we are seeing that we've got an international association now for spatial transcriptomics in plants, and we are seeing efforts to produce atlases, which will represent, in model species like Arabidopsis, all of the genes, their pattern of expression in all the cell types through development. And what you'll be able to see is a video where you can walk into the plant and look around at the genes expressed in all the cells and play that video forward to see how that gene expression changes through time as the plant grows, and then rerun it in different environments to see how the environment does that. Now, all that data has to be collected, but people are setting out to do that for model systems. So we'll have enormous resources that will bring out the dynamic nature of gene expression in plants at the cellular single cell level, and that's going to take us to, I think, a completely new level of ability to understand plant biology. but also, of course, to breed crops for particular purposes.
Thanks, Robert. So can you see the cleavage of transcripts by microRNAs? That's Peter Waterhouse.
No, I don't. We haven't looked at that yet.
Another question from Philip. Which is your favorite crop for research?
I think it has been rice, and I think it probably will continue to be rice because there's so much to do in rice, and we have now so many resources. But I think you know, in a more complex case, I think the other big focus has been sugarcane and will continue to be, because it is an important crop locally and globally, and we are really hoping to make a big difference to sugarcane in the next few years with some of these technologies. And so I think there's lots of crops we need to focus on, but those are two really important ones.
We have a question. Thanks, Robert. We have a question about spatial transcriptomics. How much single cell and spatial sequencing do you envision that will occur in the plant space in the coming year or two?
A lot. We've just seen in the last few days new projects being funded by industry in this space. We will be using spatial transcriptomics in the new research hub extensively to understand when we're attempting to modify a composition of biomass, how we can do that in a tissue and cell specific way so that we can optimize the performance of the plant while modifying its composition. So we will make extensive use of, in that case, transcriptomics in things like sugarcane and sorghum to understand how we can target the editing of genes in a cell specific way. When we're seeing the sugar industry backing this for understanding disease resistance, again, by looking at gene expression, in that case, I imagine in response to pathogens, and looking at that at the cell specific level, which hopefully will be much more informative in coming up with ways of combating those pathogens. So I think we could see it be the dominant activity in a lot of plant science in the next few years because of the power of the technology, the fact it is so data rich, and the availability of a range of competing technologies that work in this space.
All right, one last question. Thank you, Robert, for the amazing talk. Could you talk about how you use genome sequencing data, in brackets transcriptomics, to predict the biological function of the genes?
There's many examples of that, and I think the way we do that is look at the transcripts in different tissues sometimes, but largely look at the transcriptomes of genotypes that differ for the trait. So if you can look at the transcriptomes of a great example would be in wheat, where we looked at the transcriptomes of developing wheat genotypes, and we're able to identify transcripts that are differentially expressed in those wheats that produce high flour yield when you mill them, and others that don't. And what we identified is a protein that encodes a cell adhesion protein that's involved in cell binding. And what it demonstrated was that down regulation of that gene led to a seed that's more easily broken in a flour mill. So there's a classic example where differential expression analysis identifies a gene that has an obvious functional association with the trait. We've got, I think, many examples of that where, again, it's a matter of coming up with sets of genotypes that differ for lots of things, but differ specifically for the trait and looking at the association. And then obviously you want to confirm that by modifying that gene and confirming it has that function. But transcriptomics is a very powerful way to identify the genetic basis of a trait, because in some of those cases, The other thing that really is confirmatory is that the transcripts that are differentially expressed just happen to map to the major QTLs for the trait. So confirming that the gene level data and the transcript data is reinforcing the argument that you've found the right candidate genes to explain the trait. So I think there are just many examples of people doing this in terms of understanding disease resistance, understanding really any trait that you can find a set of genotypes with and without the trait at different levels. Very informative technology and relatively easy to analyze.
All right, thank you, Robert. Thank you, Robert, for an amazing presentation. Excellent. We've learned a thing or two, a lot of things actually. So could you please give me the next slide? So for the sake of time, I don't want to keep you away from lunch. The next QAAFI Science Seminar will be on the 26th. Will be by Dr. Bradley Campbell. He'll be talking to us about analysis of opportunities and challenges regarding genetic improvement in emerging Australian agricultural industries. That will be on the 26th of November from 12 to 1, same time. Next slide, please. Next slide, Robert.
Not working again, it stopped.
Oh, all right. I'll just tell everybody thank you for attending the seminar today and making time. And if you haven't already signed up to our seminar invitation list, please go to the QAAFI Science Seminar website and register there. So thank you again, and thank you, Robert, for a great presentation.